Okay, students, so we will talk about the Golgi complex now. The Golgi complex was discovered and named after Camillo Golgi during an investigation of the nervous system, and he named it the internal reticular apparatus. Now, certain important cellular functions such as biosynthesis of polysaccharides, packaging, that is compartmentalizing of cellular synthetic products like proteins, production of exocytotic, that is secretory vesicles, and differentiation of cellular membranes occurs in the Golgi complex or Golgi apparatus located in the cytoplasm of both animal and plant cells. Okay, so basically, this Golgi apparatus, I had already introduced you to this in the lecture to endoplasmic reticulum, where I had told you that Golgi is first of all, it is actually called the traffic police of the cell because it directs all the traffic of the proteins that are being synthesized by the ribosomes to different sites of their utilization, that is to the different compartments of the cell and or to the cell exterior where they are to be secreted and then transported. So the Golgi actually packages all those cargo. Like suppose you have to send somebody a gift or a parcel, then what you do is you just pack it and add an address tag to it. So in the same way, right, the Golgi plays a very important role in protein sorting, right? And then I told you it also participates or and completes the final steps of glycosylation, that is addition of these carbohydrates to proteins. These carbohydrates that are actually added to the proteins by the in the Golgi apparatus, these only actually acts as differentiation tags or the address tags that the trans Golgi network reads and then actually directs those proteins to their proper destination or site of utilization, that is to that particular cell organelle. And then, of course, if we talk of, say, endocrine cells that synthesize and secrete hormones that go and act on a different type of cell or tissue, right? So those hormones are actually packaged and in the form of secretory vesicles and then sent to the cell surface from where those vesicles they fuse and that hormone is actually released and transported through blood to the particular site of action. So all these things, that is sorting of proteins, then different modifications in proteins in terms of their glycosylation, then addition of these address tags. Okay, some carbohydrates that are added to proteins in the endoplasmic reticulum, they are again also trimmed in the Golgi apparatus also. So you'll see it actually is the center of the cell where these sorting of proteins takes place, and it finally transports these proteins after packaging them properly in separate vesicles with with a proper direction so that these vesicles can move on the microtubular tracks and reach their site of utilization. So mainly you'll see that the Golgi transports all these proteins either to the cell surface, that is in the form of secretory vesicles, then it transports the very important membrane glycoproteins to the membrane, and it also directs and actually you can say all the proteins that are or enzymes that are being utilized or are present in the lysosomal lumen. So you'll see three different type of vesicles being directed to three different directions from the trans-Golgi network, that is to the plasma membrane, lysosomes, and the secretory vesicles that will take that cargo to the cell exterior. So again, we'll learn about its mechanism once we'll talk of vesicular trafficking in eukaryotic cells. But for now, you should know that Golgi plays a very important role in the modification and direction of proteins to their proper site of utilization. Now, the Golgi apparatus is found in all eukaryotic cells. The exceptions are prokaryotic cells like mycoplasmas, bacteria, and blue-green algae, and certain eukaryotic fungi, sperm cells of bryophytes and pteridophytes, cells of mature sieve tubes of plants, mature sperm, and RBCs of animals. These are devoid of the Golgi apparatus. Again, like the endoplasmic reticulum, their number, their size, their location varies from one cell to another. Again, it totally depends on how active a cell is in synthesizing a different type of proteins, how active it is in protein synthesis, right? So, if more proteins are synthesized by a cell, or very readily proteins are synthesized by a particular cell, then of course their sorting and their modifications will also be raised, and the function of Golgi apparatus is more there, and hence their number and their location varies accordingly. So here you can see the Golgi apparatus. Now if you just take a look at this picture, it actually just looks like the endoplasmic reticulum, because it is even said that earlier, when these used to be a single organelle, right? And it is said that this Golgi and ER, I mean, they have all evolved from the nucleus, and that is why they have very similar, I mean, composition of their membranes, right? So structurally, it represents or looks like the endoplasmic reticulum. Then I had already told you that it has three regions: cis, medial, and trans. The cis region is the receiving side of the Golgi apparatus that actually receives all the proteins from the endoplasmic reticulum, where it then sorts all the proteins, okay, whatever proteins belong to the ER, it sends them back, and the other proteins, they move to the medial and the trans region, where further modifications in the proteins are done. So here you can see in this diagram, this transport vesicle from rough ER is being received at the cisternal side of the Golgi. Then you can see the trans Golgi region. The trans phase of Golgi is called the shipping side of the Golgi apparatus, where you can see the new vesicles being formed, and these vesicles actually carry these transport vesicles. These carry all the cargo, that is the proteins and the lipids, all both membrane and luminal proteins, being packaged and transported to different destinations. Now, so the morphology of the Golgi apparatus actually resembles that of ER. The Golgi apparatus is morphologically very similar again in case of both plants and animals. However, it is extremely pleomorphic. That is, in some cell types it appears compact and limited, while in others it spreads out and appears reticular or net-like. Its shape also varies depending on the cell type. It appears as a complex array of interconnecting tubules, vesicles, and cisternae, just like the endoplasmic reticulum. Again, the simplest unit of Golgi apparatus is the cisterna. This is a membrane-bound space in which various materials and secretions may accumulate. This is actually the receiving side of the Golgi. Numerous cisterna are associated with each other and appear in a stack-like lamellar aggregation. A group of these cisterna is named dictyosome, and a group of dictyosomes make up the cell's Golgi apparatus. So the detailed structure of Golgi again is can be divided into flattened sac or cisterna. Okay, or are these are about 1 micrometer in diameter, flattened, plate-like or saucer-like, closed compartments which are held in parallel bundles or stacks, one above the other. In each stack, cisterna are separated by a space of 20 to 30 nanometer, which may contain rod-like elements or fibers. Then the tubules form a complex array of associated vesicles and tubules with a size of 30 to 50 nanometer in diameter, and they surround the dictyosome and radiate from it. The vesicles, of course, as you have seen in the diagram, they are approximately 60 nanometer in diameter, and these vesicles that arise from the Golgi, they are of three types. First one are the transitional vesicles, okay, which are formed as blebs from the translational ER to migrate and converge to cis phase of Golgi, where they coalesce to form the new cisterna. Okay, so these are called the transitional vesicles. Then we have the secretory vesicles, which are varied sized membrane limited vesicles which discharge from margins of cisterna of Golgi, and they often occur between the maturing phase of Golgi and the plasma membrane. You'll see these vesicles in between the cytoplasmic part that covers the space between Golgi and the cell surface. Then last are the clathrin coated vesicles. These are spherical and about 50 micrometer in diameter, and they have a rough surface because of this clathrin coat. Okay, they are found at the periphery of the organelle, and these are the vesicles, right, which are actually delivering the membrane and the lysosomal protein. So these secretory vesicles actually they are not always fuse to the cell exterior, and they release the cargo extracellularly. Okay, they are just formed with those packaged, you can say, neurotransmitters and hormones, and they just lie near to the cell surface in between the space between the Golgi and ER. So whenever there is a stimulus, they will release the cargo to bring about a biochemical response. Okay, so transitional vesicles, they actually form the Golgi cisternae. These secretory vesicles, again, they are carrying different proteins that are to be released to the cell exterior, and the clathrin coated vesicles, they carry cargo that are directed either towards the plasma membrane, which are actually the membrane proteins, and the lysosomal proteins. Again, we'll learn the formation of the clathrin coated vesicles, how cargoes are packaged in them, and how they are delivered. Again, once we'll talk of vesicular trafficking in eukaryotic cells. Now, in the last, let's talk of the functions of ER. So, modifying, sorting, and packaging of macromolecules for cell secretion. Okay. Again, just like the ER, the Golgi modifies, sorts, and packages proteins to be distributed to different parts of the cell. Then, proteoglycans and carbohydrate synthesis. That is, these include the production of glycosaminoglycans, right? The heteropolysaccharides, long unbranched polysaccharides, which the Golgi then attaches to a protein synthesized in the ER to form proteoglycans. Once you learn about the extracellular matrix, you'll see they form a very important component of the ECM in animal cells. So these are actually synthesized in the Golgi apparatus and then transported through secretory vesicles to the cell exterior. Then again, in animals, Golgi apparatus is involved in packaging and exocytosis. Plants, if we talk of, Golgi is mainly involved in the secretion of materials of primary and secondary cell walls. Once you learn about the molecular control of cell division and cytokinesis, there you will see how the plant cell wall is formed in newly forming cells. These Golgi vesicles, they carry all the polysaccharide content to the equatorial plate. Okay, we'll learn about their mechanism there. And then during cytokinesis, either in mitosis or meiosis, the vesicles originating from the periphery of Golgi apparatus, they coalesce in the phragmoplast area to form a semi-solid layer, which is called the cell plate. Okay, I'll not go into the details of cell plate formation here because we'll talk about this once we learn about the molecular control of mitosis. Okay, so this is all about the structure and the important functions of the Golgi apparatus. Thank you.